So I'm up here talking about this, but you've all seen Brian already, so he's going to help quite a bit. And then Deborah's over there on the listeria table, right? Listeria? So Deborah's the infection control nurse at Caboolture Hospital, and really most of the work that's involved in this outbreak is she's responsible for. So anyway, we're going to talk about a little outbreak which was unexpected, but I guess expected outbreaks are to be avoided in healthcare. So Caboolture Hospital is a little 265-bed hospital, somewhere around there. It's sort of midway between the Sunshine Coast and Brisbane, which sounds very nice, but it's in an area of really low socioeconomic, poor determinants of health. So as a doctor, you get a lot out of working there. There's a rich substrate. And the drainage area for Caboolture Hospital is this enormous, so this graph here is the entirety of Metro North, which is this HHS that we all work in, or that people from here work in. And Caboolture Hospital drains everything going north all the way up to sort of Blackbutt, and so you see lots of interesting zoonoses and rural-related diseases. And because it's not a quaternary or tertiary centre, we're all very happy and lovey, and we all work together, and so we have this motto of caring together, which is all very nice. So this outbreak, there's a little bit of tongue-in-cheek there maybe, but certainly Deborah and I, we love working together. So this outbreak occurred in the special care nursery, and it's I think a level 2 nursery, so it only looks after babies that are greater than 32 weeks and over 1,500 grams, and it's got 12 beds, or at least it was designed for 12 beds, but because of the typical pressures and so on, bed 7 turns into 7A and 7B, and bed 8 or incubator 8 turns into 8A and 8B quite commonly. So overcrowding within the unit is the norm, and it's got the usual sort of infection control structures in that the babies that are there's surveillance screening for multi-resistant organisms once a week. And babies that come into the unit and leave the unit are also swabbed. And they have about 30 admissions a month into special care nursery. Most of those are directly from the birth suite, where they do about 150 births a month. So a very fertile population that Caboolture drains. The index case was born after a Cat 1 caesarean section at 34 weeks and admitted directly from birth suite into special care on the 24th of August 17. And 5 days later, on routine swabs, the laboratory reported a Klebsiella oxytoca ESBL producer in brackets. And so I can see Anna over there, Anna Hume on the listeria table. So what do you make of that? And is that important from a micro point of view? Because Deb called me and said, what do you reckon? I was like,
yeah. And so, obviously, in terms of what neonates are colonised with, there's a bit of paucity. We don't tend to do research on neonates and their microbiomes. However, we do know some general principles about what they're colonised with. And also, this is a threat level, which means that their guts are typically compromised. So, we wouldn't typically expect this to be part of their normal flora, and we would also be prepared for basic infection.
And Graham, as the boss of the laboratory, what does this comment mean? ESBL, Klebsiella oxytoca. So you've got to take an infection control sort of attitude towards this organism in special care. So is this, is it truly an ESBL, or could it be anything else?
Well, it's possibly could be a hyper K1 could be. So that's the commonest cause of that sort of resistance in oxytocin. Now, I presume in it's my lab that they will recognise that issue and sort of out the easily field produced by doing other tests. I hope that's...
Well, just the phenotypic test that was done. The phenotypic test, the cetraxone, chloronic acid was consistent with an ESBL.
Yes.
So I think that can be, wrong in the case of a K1. And just for everyone that's not a microbiologist, we worry about ESBLs because they have, this plasmid that Patrick was talking about earlier that may, enable gene transfer to other organisms. So they have a specific sort of, risk profile in themselves. With Klebsiella oxytoca, often have this overexpressed chromosomal gene, which can result in falsely looking like an ESBL. So when I was called about this, I sort of said, oh, well, it's a bit interesting that, weird things happen, and let's just ignore that for the moment. Would anyone have done anything different? Obviously, they were put into precautions, and we all wore aprons and killed some turtles and so on, classic. But would anyone have done anything different?
Nope. Do some screening? Yeah.
Yeah. Good. All right. So obviously this is an outbreak talk, so there's going to be a next case. So we were hoping we were going to blame the next case on Raw Brisbane, but unfortunately it didn't work out. So this next case, again, born via caesarean section on the 15th at 35 and 3, and was back transferred from Raw Brisbane to special care nursery. And they actually had 3 sets of swabs done, and so the first 2 sets of swabs were negative. One was performed as the intra-hospital transfer into the unit, and the next was the routine, everyone gets a swab on Monday type swab. And then on the 26th, so, a week down the track, they came up with a Klebsiella oxytoca ESBL producer. Does anyone want to, what would people do about this now? So these organisms have been isolated, a couple of months apart. A month apart. There's no overlap in the baby's bed stays.
What about their equipment use?
Well, yeah,
Overlap in the equipment use?
there will be because the incubators are reused.
For those nurses bearing false claims?
Well, that's a whole can of worms. I think actually, the special care nursery are super dedicated staff, and they're really, they're really careful, and they don't work in special care nursery because they want to have quiet time. They're really committed. And so, in fact, during this outbreak, we did a lot of unit monitoring, and their compliance with sort of basic infection control procedures is pretty good. But that does come up later, so we'll talk about it. So I guess the key thing here is that there's no clear epidemiological crossover with these organisms, or the babies, or the mothers, and all that sort of stuff. We're a little bit lost. We'd obviously increased some cleaning in the rooms, and we'd thought about some environmental cleaning. But one key thing here, and it may differ depending on which labs you are, is that you have to call the laboratory, in which case I called Patrick. Because the standard workflow, at least when this was going on, was that if you take a swab, a surveillance swab, and it grows something, that's the end of the story in the laboratory. And the isolate goes in the bin, and the swabs go in the bin. And so the key thing here was I called the lab, and the lab was, from this isolate onwards, stored the organism on ice. And so later on, we were able to come back to it, if at all necessary. And at the beginning of an outbreak, you don't actually know it's an outbreak, so it's important to sort of try and think ahead when you see slightly unusual things and start to generate some data for later. So case 3, so that last case was in September. Another month down the track, lots of screening going on, sort of furious infection control practice, another one. So this time, can we just, why don't we, from this table, is there any suggestions of what we should do at this point?
I think at this stage you're probably thinking it's a definite outbreak, and
yeah,
you've got 3 cases.
Totally, yeah. So this is where we, despite the lack of epidemiological crossover, we declared it an outbreak. And so the next table, I don't know what organism you are, but once you've declared an outbreak, what are the important steps? What do you need to do in that response? What does an outbreak response look like? Janice, I'm pretty sure you might have done one of these before. Yep. So you need to involve the laboratory, you need some clinicians involved, you need the infection control people. Super importantly, you need to involve the unit. So if it's going on in special care, you need the NUM there, and you need to have a little meeting with the work staff, and you need the executive, and especially babies, really touchy-feely sort of subject. Got to be careful. So we're going to talk about a response. can we get everyone maybe just to sit down in their little tables and come up with a little outbreak response at the moment, just a clinical response to this situation? We'll give it just a couple of minutes and then come back.
I've actually done this many times before just simply pretend I was and I sat there for about 2 hours and I just the flow it's that's like that. There's some sort of
All right. How'd everyone go? Has everyone got a response sorted out? Yeah, well, actually start over here. Why don't you tell us about your response? What you want to do?
Investigate.
Yeah.
Isolate the patient. Implement precaution. Transmission precaution.
Yep.
Collecting samples, including markers and protocols. When a patient was just that, we do take samples.
Very good. That's good. So we've got doing samples of babies and mothers. I think you said epidemiological investigation of beds and that sort of stuff. And then case management, obviously. I guess defining a clear case definition is important as well. And for us, it was going to be just a ceftriaxone-resistant Klebsiella oxytoca. So we did those things. We did some furious environmental sampling, and we obviously had a communications program with nursing and exec and the laboratory. I think talking to the laboratory is so super important, and things often get lost in the 2 inches of space on the request form. So I think having a laboratory liaison is so important. Obviously, we started giving empirical treatment advice to the clinicians at that point, that if they had, a febrile neonate, which is basically all of them more or less, what to do with them. And unit observation, so just basic infection control epidemiology, just stalking around the unit regularly, camping in there, watching what people are doing, and you come up with unusual stuff. So we increased our swab surveillance, so we were swabbing people every 48 hours. The turnover within the unit is high, so babies don't stay in there necessarily for a week. They're out often within a few days. So if you're trying to catch an epidemiological focus, you need to start generating a bit more data. And so between August and January, we had 6 cases. 5 of those that we had sequence data on, or that we had the organism from, and you can see their admission and discharge dates. Apart from 5 and 6, which only overlapped by a day, all of those babies were in and out, and the mums were in and out of the hospital without overlapping each other. So it was a bit of a mystery. This is just case 1. So again, this demonstrates sort of what you imagine would be the logical thing to do in a unit from an infection control perspective, but what actually happens in the unit is completely different once you start, wandering around and following people. So case 1 started in bed 3, and the next day went to bed 7. And the next day went to bed 5, then 12, then 11, and got isolated with pleboxy, so ended up in isolation. Then bed pressures and stuff, because we had more than one there, came out of isolation and they went back in again. So there's this daily basis, and trying to form an epidemiological map with that many bed moves is difficult. We talked with Special Care Nursery about this, and the drivers behind this sort of bed moves and so on are, things that you don't really conceptualize when you're thinking about it from an infection control problem, and they move the babies on a sort of cyclical pattern through the unit based on how acute they are. So this is the number of beds, the number of bed moves these babies had. You can see we're over census there a bit with case 5 and case 6. So there's a little bit of bed overlap, but epidemiologically the babies didn't overlap very much. So at this point we'd had 42 separate samples sent away, and we found nothing terribly of interest except for a Klebpneumo in a sink, and an E. coli ESBL in a baby bath drain. And at this point, we got a report back from Brian. So that's included in your handout, or at least a slightly amalgamated one of that. So I was hoping you can have a look at that now and have a read through the report. This will be our next breakout thing. And if you guys discuss between yourselves what the report means, and we'll try and work through what it means together. Brian, maybe if you can help and the matrix.
So it should be under 25.
everything there.
What do you suspect clinically?
And the number 6. Yeah. We do, I'm thinking of
So it's interesting.
So this It actually shows.
So that's good, everyone. So everyone's had an opportunity to work through that first sheet, I presume. That's good. So why don't we'll start with the middle table. We can start with Crispin's table. I forget what organism he is. You're MRSA. So can one of you guys just tell us about what the basic elements of the report are? Yes, excellent. Tara, what's the next?
It's unlikely that they're all coming from a single source, so they're probably independent introductions into the healthcare facility, but there may not be a single source for their...
Was that multiple iron, or?
That's what we thought. Results.
Yeah, so all the organisms were related at the core genome level, is that correct?
So
they're novel sequence topic?
Yeah, we couldn't sequence type them, so it was novel that we didn't see those allele combination before. And based on those SNPs, distance that we saw and everything, we thought that they were related, but broadly, and possibly from a kind of common but diverse environmental reservoir in the hospital somewhere.
Okay.
So if it was Crispin being the index case, and he gave it to you, and he gave it to you, and so on, we'd expect in that table of matrices the number of SNP differences between Crispin's organism, your organism, to be less than 30 or 6.5 or whatever the arbitrary number we want to...
Yeah, so in this case, I think these are about 6 megabase genomes, so 30 SNPs would have been our cutoff for cluster.
So there are multiple transmissions in between.
Going to the next person. And it's not necessarily consistent with everyone catching or acquiring this organism from a single source that has a single pure growth either. Because again, then you'd expect it to have a similar sort of SNP pattern.
Or just really well established, I guess.
So we were thinking about an environmental source, somewhere where the organism has been sitting and breeding and hanging out and mutating and acquiring lots of different subpopulations of the same organism. So we needed to go hunting further for an environmental source. We'd already just exhaustively. Deborah can tell us, what did we do in special care? Well, we didn't have sledgehammers, but...
We started clinical project, and myself looking there and looked for any wet sources that we could find. So looking at lotions and potions, and basically swapped anything that we could find that we had to report from partly all the way down to the environments, what we there. And also had lots of conversations with the special care nursery staff for them to give us ideas of where a possible source to be. And so they were very forthcoming with all sorts of ideas. A lot of these were their sources, but they even down to swapping the wipes and things like this, because they're concerned about the wipes. And I think also just that direct observation we were talking about before, actually watching what was going on. I think that was the one I was talking about. I think that was that direct observation, which wasn't actually the pure nursery at that time, was how we came to discovering sources.
So we're 3 months into this outbreak now, elbow deep into it. The nurses are freaking out. They think that they're carrying it. There's just this huge sort of anxiety within the unit. The special care nursery, they're super dedicated nurses, and they really care about their tiny babies. And they were freaking out. They were anxious. It was really a stressful, hard time for them. And this report coming through illustrating that it was likely that it wasn't person-to-person transmission via healthcare workers' hands was really, psychologically important for them. It really helped the nurses at that time. So, Graham.
So, a question perhaps for Brian. So, the resistome is got there is identical, and I guess we don't look at resistomes with particular osteoporosis every day, so I don't know how common that resistome is, but isn't that a bit of an indication of either mobile element transmission or, or some relatedness? And what about the rest of the accessory genome? Is there any other evidence there?
So it's for those reports, maybe we look at resistance chains in terms of the accessory genome, but that's likely, as you said, that's most likely part of the plasmid. That plasmid is just, endemic in that population. So either they're transferring between or they both have it from a common progenitor, which is stable. It's likely that it's stable. I guess that's what is the advantage of this in the hospital setting. But yeah. So we wouldn't really tell us how we do it related. How related they work with each other.
That's good. So we'd exhausted special care nurseries, so we expanded our environmental sampling outside of special care into birth suite and maternity. We kept up the pressure in the neonatal area. We had lots more communication sessions, and again, some of these were getting a little bit heated because the nurses were so stressed out. The question of whether to swab staff came up. Early in the outbreak, the staff had been like, why don't we swab ourselves? And then this came up, and we'd sort of said, well, maybe. But that's a massive can of worms, as you can all imagine, and you have to get unions involved and have some kind of idea of what you're going to do with people once they're isolated. So obviously that didn't happen. So the next thing I wanted us all to look at was the Snip Matrix. So this is, we'd exhaustively kind of sampled around the hospital, and we started finding some cases. So we ended up getting nine cases, which and a bunch of environmental isolates as well, all growing in the CSBL Klebsiella oxytoca. So I want you to look at this larger Snip Matrix and start to, you can use a pen or something to start to put a ring around cases and samples that are likely related, and come up with some ideas and see if we can get a source from that. So we see, the source mostly always ultrasound gels, barrier creams and stuff. Some all sorts of stuff to allow. You can see there's clearly a here. So it's in case 2, case 4, case 5, and 3. These are all related with environmental source too. So we're looking at the environmental source and the environmental source. That's good, everyone. So I think everyone's on track there. So from another random table, I'm picking on the front. So right, you guys on whatever table you are. Klebsiella, how appropriate. Serendipitous. Because, so you guys, what do you think is what's coming out of that neighborhood?
There seems to be lower numbers between all of the cases, so in the vertical column, and case 789, and the only environmental case that's got the similar no numbers is environmental 4.
Yeah, that's great.
So we want to know where that is.
Yeah, excellent. So that's exactly right. So again, so this is later on down the outbreak, and there's more data, and I think Brian will tell us why some of that SNP distance has reduced so much. We just mentioned this environmental source 4, and these cases here being so closely related, the SNPs were only in single figures, and again, case 7, 8, and 9 again being associated with environmental source 4. So outbreak for this came from just watching what was going on in the maternity unit, and we expanded our swabbing all around maternity and birth suite, and we swabbed this multi-purpose room. We swabbed this drain here, these are both baby baths, but they're no longer used. You can see there's a washing machine and a dryer, and in these cupboards they're full of old dirty vases, which the flower team, so hospital volunteers, went around the hospital placing flowers around. So the first positive we got was from this drain, and Deborah staked the place out and noticed the flower service using the room, and so this is the little basket that the lovely volunteer service wandered around the hospital. Cleaning out vases, changing the flowers and that sort of stuff. And so we swabbed a whole bunch of stuff from within that basket, and we got a positive from this bottle of detergent. So at that point we closed down the flower service, which is a bit sad really, and we shut down that multi-use area, and we sort of decommissioned it. And we went looking around the hospital for other bottles of detergent, and we found a whole bunch of detergent bottles which were positive for this Klebsiella oxytoca ESBL. And environmental source 4 was the detergent which was in the milk room of special care nursery. So this is special care nursery here, and you can enter here, exit here, and enter here. And the nursery room here, that's actually the milk room. And so this is where all the mums went to pump milk, essentially. And the whole purpose of these babies being in special care was usually because they're premature and they had low birth weight. And so all these mums are just furiously pumping, trying to get some nutrients together to give to the babies. And they were all aware of this outbreak, and so all the mums were just being super diligent with washing their pumping equipment after use. And the pumping equipment has 24-hour disposable suction caps for the breasts, And so you'd pump a couple of times a day, and eventually, after each pump, you'd wash it up with detergent, take a break, and, pass out or something, and then get back to pumping, trying to get these babies heavy. And when I called all the mothers that I could get in contact with, and they all reported that they were pumping for their babies and were meticulous in washing up their pumping equipment afterwards. So outside of special care nursery, quite remote from it, is the laundry, where we got our first break, I suppose. And then these bottles of detergent were all over the hospital in various places, on different levels and in different units, and not in natural places where you could imagine someone going and grabbing a bottle of detergent, wandering back to their own unit and leaving it on the sink. So we found 12 detergent bottles around the hospital that were positive, and including detergent in the adult ICU, but I guess none of those were pumping, so they escaped, and in the paediatrics room. So I might ask Brian to explain this slide. Would that be all right, Brian?
Yeah. So this is, I guess it's kind of we've got a minimum spanning tree. It's a simplistic way of viewing the relationship between isolates and the outbreak. So you can see we have two clear groups. We have one in orange or red at the top, and then we have these blue groups at the bottom. Again, similar to earlier, everything within a circle is identical at the core genome level, so 0 SNPs. The SNP distances are kind of indicated on the branch with the numbers. So in our blue group, we have our patient isolates, and in our red group in the top is our environmental isolates. And you can see the cluster distinct from each other, except for that one environmental isolate that Joe pointed out, which is actually identical to 2 patient isolates. What's interesting about this for us in the patient isolates is that they all radiate out from this central group, which includes this environmental isolate, suggesting to us that it's this group in the center is probably the source of the outbreak. But you've got to be careful when you're interpreting these trees, because they don't really represent the actual kind of directionality properly. So it's kind of, it gives you an indication of where to start for result.
I think super powerful in this diagram is the fact that associated with that environmental source, there are 2 patient cases, and those patient cases were patient 1 and patient 10. So they were 6 months different to each other, and despite that 6 months of time, had exactly the same core genome. So it's not like it would be fantastically lucky, I would say, for the organism from case 1 to have undergone 6 months of genetic mutation, in which case it comes back to exactly where it was 6 months ago at the core genome level. It seems improbable. Would that be right, Brian?
Yeah, I'd say we'd be looking at almost everything being identical in the core genome level, but the fact that we have that one environmental isolate in there, and I think that isolate was collected just a couple of days before that final patient.
Yeah. Very good. So in the hospital, all the detergent comes from this, it's not a 44-gallon drum, but I'd like to think it was. But they're big 20-gallon drums of concentrate, and we go through a bottle of concentrate about once a month. Is that right? Yeah. So all the cleaners around the hospital, it's again the things you find out when you start actually wandering around the hospital and talking to people. So all the cleaners, when you want some detergent, or what's supposed to happen anyway, when you want some detergent, is you go up to the big detergent thing, you pour a little bit in, well, not a little bit, but clearly described amount into the bottle, and then you fill it with water to dilute it, and then you take it back to where you want to go. But clearly that dilutional kind of accuracy wasn't necessarily being followed. And additionally, the practice of completely using the bottle of detergent, washing it out and drying it was, a new concept to all the cleaners. And clearly, if you're not actually thinking from an infection control point of view, that seems like a ridiculously labour-intensive sort of action to do from a cleaner's perspective. If the bottle of detergent's a little bit low, you just go and top it up. But that probably, what I think is that probably allowed propagation of the organisms after contamination from a single central source in the bottles over time, and allowed this propagation of this organism over 6 months. We tested the bulk detergent, which was negative, but we go through about a bottle of bulk detergent once a month, so I suspect potentially we had a contaminated bottle of detergent at some point. Again, I'll hand over to Brian for these clever Star Wars-looking diagrams. Something to do with a mushroom.
Yeah, so this is the data that's actually on that Snip distance matrix that you're all looking for. And this is massive data that we use to construct cluster diagrams in CAPI. So this is actually just an output vector from CAPI. And it's a bit static now, but it does look a lot better when it's online. But what I did, I just showed that basically we have two different clusters of isolates. So we have the environmental isolates and the patient isolates, and they are related to each other. So they're in, the environmental isolates are within 15 or so core genome SNPs of the actual patient isolates. And where the arrow points in the top one, that's the environmental isolate. So just in this
So
diagram,
that's environmental source 4, that one.
yeah. So in this side, we just click, we just selected that particular isolate, and the color coding shows you on the heat plot here the approximate SNP distance between those isolates. And then on the other one, we're just on the same line, we selected one of the environmental isolates. So you can see that within each group, they're pretty closely related, but there is a bit of a distance between them, except for that one environmental isolate.
Yes.
At what stage during this whole process did you reclassify Oxytoca to the image?
Oh, well down the track, and we'll talk about that. So, yeah, I'd like to say that we looked at it as classic Michiganensis. Yeah, that wasn't the way it went. So we wrote this up, or are writing this up, depending on how you look at it. Brian and Pat are involved and done a lot of the work in that manuscript, and we submitted it to. It wasn't. What was the genome?
We submitted the GenBank.
GeneBank, of course. The GeneBank looked at our sequence data and actually said you've got the species wrong, which is reasonably embarrassing. But I think Brian's going to talk a little bit more about why that isn't embarrassing, which is great. This is a great diagram that Brian's group have produced. Each of the black lines is a patient. And you can see that really up until January, there was no overlap of the babies in time. This is time along the x-axis. All this time, we'd been furiously environmentally sampling, and this is where we got our break when we expanded the sampling out to maternity as well. And this is essentially where we started looking at detergent bottles. And a month after we'd eliminated all the detergent in the hospital, or changed it anyway, to a different practice, we had no more cases, and we called the outbreak over. So, Brian, can you explain why it wasn't embarrassing that we?
Yeah. So this is the first time I've ever heard of Michiganensis as well. So when we submitted our genome, and now it's there's a big issue with GenBank, which has been, I think, Caitlin alluded to earlier about the quality of submissions on GenBank, and we know this ourselves, and then we're complicit in it too. But nowadays, when you submit a genome to GenBank, they automatically screen it to taxonomy. They do some taxonomy profiling themselves, and they use a more sensitive program than we use. They use something called average nucleotide identity, which is you look at the coding regions of the genome and you compare them to other genomes, and you get back an identity to whatever reference. And they came back as ours was much more related to Michiganensis, about 98% identical to Michiganensis, but only about 95% identical to Oxytoca. So these two species, they're really similar. I think they're nearly 95-96%. I think they're really hard, and you can't distinguish them by 16S rating, so they're nearly identical. So we looked up to see what was going on and helped them did some phenotypic testing, and even just based on the urease production, you could see that they were actually correct. So we didn't trust them when they said it. So but we said
Weird example
Big
of
brother.
genome sequencing sent us back
Yeah.
to old-fashioned.
so. But then we decided to go a little bit step further because we weren't very happy with the outcome about how we made this mistake. So what it is, we took some representative pneumonia genomes and some just basic Klebsiella genomes and all the oxytocas and all the Michigan-Genesis genomes we could find online, and we decided to build a tree out of them. And what we found is broadly, you can see here, so we have all the breakdowns. We have the varicella, the pneumonia, these unknown species, and then in the blue here we have everything that's up in NCBI that's taxonomically profiled as oxytoca. And everything over this side in the yellow is Michigan-Genesis. But we can see here there's a hell of a lot of blue inside Michigan-Genesis, and these are all oxytoca isolates that have been misclassified.
So all these blue excision numbers are...
And here's our MA2255, that's in red, that's where our genome sits. But what was quite interesting is that over in green here, you can see this is the type strain on NCBI for K-oxytoca. And that's actually in Michigan-Genesis as well. So it's actually their fault that we were wrong.
That's right. Genome to the rescue again. It's great. Yeah. All right. So, and I guess, our diagnostic medium for speciating these organisms we rely on completely, but it's only as good as the database, I guess. And I don't know, was Michiganensis in the MALDI-TOF database, do you think, Pat?
No.
All right, great. That's good. So conclusions. So I think, what I learned out of this outbreak anyway is that the super important thing to do is to maintain communication with your laboratory. And right early at the start of an outbreak like this, I'm a clinician, I'm not a microbiologist, so I don't necessarily understand all this crazy indole production stuff. But being in communication with your laboratory is super important. And if you do talk to them, in my experience, they've been happy to store isolates, and later on down the track, that becomes really important. And thank you, anyone.